this recording, we'll discuss the meninges and ventricles of the brain. Your brain is really important. It does lots of nice things for us, so we try to protect it any way that we can. So in addition to the skin, the periosteum, and the bone that cover the brain, we also have three protective membrane layers. We call these cranial meninges, and they have funny names. You've got the dura mater, the arachnoid mater, and the pia mater. And these meninges help cover both the brain and the spinal cord. And so these terms will come up when we mention the spinal cord in later lectures. They also contain blood vessels that help with the creation of cerebrospinal fluid that we'll go into a little bit more detail about here soon. And it helps secure the brain to the skull. So we don't want your big old brain just kind of jiggle around inside your skull, because we don't want it to get damaged or bruised or anything like that. So we kind of hold it in place with these meninges. So if we go superficial to deep, the first meninge that we would come across would be your dura mater, your tough mother. And this is actually a double layered structure that contains the dural sinuses. Now in this context, your dural sinuses, these are large veins that lie in between the two layers of the dura mater, and they collect blood that is draining from the brain, and the drained blood goes to the internal jugular vein, and eventually that blood makes its way back to the heart. Now, if you remember, hopefully you do, the longitudinal and the transverse fissure. Okay, these are where the sinuses live. Okay, so your longitudinal fissure contains your superior and inferior sagittal sinuses. Okay, so you can see superior and inferior ones here. Okay, and then your transverse fissure holds your transverse sinus. There we go. You can see just how large these veins are. Okay, these other little blue ones, these are like quote unquote like normal sized veins or even venules. These are giant. Okay, but you remember how much blood we sent to the brain? That should make sense, because whatever goes to the brain has to come back from the brain. Okay. Now, if we keep going deep, next we would hit the arachnoid mater, the spidery mother. This contains your subarachnoid space. You're likely to find thread-like extensions here that connect the arachnoid mater to the innermost layer, the pia mater. The arachnoid mater itself does not travel down into the sulci. Okay, the pia mater does. So the arachnoid mater, it's really skinny. It's a little hard to see, but it's this thin green layer. Okay, it travels into the fissure, but it does not travel into the sulcus here. But this little red line, everybody see this thin little red line? If we travel back, that's the pia mater, the innermost layer. Okay, so that's a big difference there. Arachnoid mater does not go into the sulci, the pia mater does. Now, the subarachnoid space that we just mentioned, here's your subarachnoid space, this region right here. This is where you're going to find cerebrospinal fluid, and you'll have additional blood vessels to help make sure nutrients are getting where they need to. And cerebrospinal fluid is actually made from the blood plasma, and so it should make sense that we have blood vessels here. Now, the pia mater, last layer, innermost layer, touches the brain like shrink wrap. Okay, it's held in place by astrocytes, which we have previously mentioned. And again, this thin little red layer does go into those valleys, those sulci of the brain itself. Okay. All right, so cerebrospinal fluid. We keep throwing out that term, CSF. This is similar to blood plasma because that's basically how it's created. We start from blood plasma. It's going to circulate around and cushion and nourish both the brain and the spinal cord. We form CSF from ependymal cells. Okay, we've mentioned ependymal cells previously in other recordings. These ependymal cells specifically are located at the choroid plexus, and we'll see a picture of those here in just a little bit. These are going to be located near the ventricles, which should make sense because ventricles contain cerebrospinal fluid. And the fluid is going to be moved by the cilia that we find on the ependymal cells specifically. We've got all these little cilia. Okay. Now, to form cerebrospinal fluid, again, we start with the blood, the plasma specifically. We have hydrostatic pressure that pushes blood out. of the choroid plexus. Okay, so we can get it into the ventricles. Okay, that way we have nice CSF filling up those ventricles. Again, we're cushioning, we're nourishing the brain, all kinds of good stuff. Okay, now the ventricles themselves, okay, develop from the neural tube, which is basically how your brain starts, really just kind of a tube, and then it turns into a brain eventually. But the ventricles are basically all interconnected with the central canal of the spinal cord, okay, and they're all lined with the ependymal cells, and then the choroid plexus specifically is where the fluid itself is produced, okay. The lateral ventricles, which we have previously mentioned, okay, they're kind of surrounded by the cerebral hemispheres. We separate the left and the right lateral ventricle by this region called the septum pellucidum. Okay, you're likely to see that on images of the brain or the brain models, things like that. Okay, now just in case you never get to see a real life brain and hold it in your hands, let's kind of give you an idea of what these ventricles look like. So from an anterior view, the big lateral ventricles, okay, which you do have two of these. You have a left and a right lateral ventricle, okay. They kind of look like sheep's horns or ram's horns. They kind of curve like that, okay. They're pretty good size. They are connected, and here's an actual lateral view, okay, so you can kind of see just how significant in size these guys are, okay. These are connected to the third ventricle, okay, which your third ventricle is this guy right here. Kind of looks like a bird's head. Okay, so your third ventricle connects to your lateral ventricle through this tiny, 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 thin little thing called the interventricular foramen. Okay, and so if we look in the front, it's just this little region right here. It's very small. Okay. From the third ventricle, we drain down into the fourth ventricle. Come back. Okay, fourth ventricle. Okay. And again, we've got a tiny little connector tube. It's called the cerebral aqueduct. Okay, we can see it from a side view or frontal view. Okay. The fourth ventricle, if you look in anterior view, it is kind of a decent sized ventricle. If you look at it from just the lateral view, it looks very, very small. So it's a little deceiving if you only look at the lateral view. And then the fourth ventricle is continuous with your good old central canal of your spinal cord, and so cerebrospinal fluid just continues down, and eventually it would come back up and circulate. Okay. Now, there is a whole pathway. Okay, and we just love it when y'all learn pathways of things. Okay. We start creating cerebrospinal fluid, we mentioned earlier, with the ependymal cells, specifically in the choroid plexus. Now, I did tell you I'd point out, so here we go. This little red area up here, and this really thin red area down here. These are your choroid plexuses. This is where we are pumping out cerebrospinal fluid, okay? From the choroid plexus, we first enter the lateral ventricles. We move through the interventricular foramen to the third ventricle. We go through the cerebral aqueduct to get to the fourth ventricle. We said the fourth ventricle was continuous with the central canal of the spinal cord. And then we kind of come back up and we would circulate through the subarachnoid space. Kind of, here we go, subarachnoid space, until we got reabsorbed by all these little things up here. Everybody see these little guys? These are the arachnoid villi in the dural sinus. Okay, and so kind of here's a better picture. All these little nubs. See these little nubs? Here's one of these nubs. Okay, this is one of those arachnoid villi. Okay, we are going to kind of reabsorb that fluid back into the dural sinus. Okay, so cerebrospinal fluid basically starts as blood plasma, and it kind of ends up back as blood plasma. Well, that's kind of interesting. Okay. Please, 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 everything underlined, make sure you are familiar with this correct order. Okay. Now, sometimes things go wrong. Okay, you can accumulate too much cerebrospinal fluid in your subarachnoid space. This would lead to intracranial pressure or increased intracranial pressure. Oftentimes this is caused by blockage in your ventricle. It could be congenital, which means you could be born with a problem. Perhaps an aqueductal stenosis. So stenosis means we have narrowed something. So aqueductal stenosis, we have narrowed that cerebral aqueduct. Okay, so not as much fluid can flow as normal. You could also acquire something, perhaps an infection, a tumor, meningitis. Any of those could also block off either partially or fully a ventricle, and that fluid's then just going to build up instead of being reabsorbed properly. If this occurs in babies, okay, if we don't really address the problem and relieve the pressure, this could lead to learning disabilities, short-term memory loss. If it gets really bad and just legitimately goes untreated, this could eventually kill the infant because you are just putting way too much pressure on the brain. Remember, the brain kind of controls, breathing and heart rate and respiration rate, things like that. So if you put too much pressure on the brain, you can negatively affect those types of processes, which could ultimately lead to death. Okay, this could, if it's acquired, then obviously, this could happen as an adult as well. But same thing could happen. Increasing intracranial pressure too much can ultimately lead to death. So we would need to relieve that pressure if at all possible. Now, the blood-brain barrier, we briefly mentioned that as well. We associate it with the astrocytes that we talked about in a previous lecture. The neural tissue within your big old brain, we've got to feed the brain, all right? So we've got capillaries that are going to bring in all the nice oxygen and nutrients that the brain requires. But these capillaries are pretty darn impermeable. So most of the things that you find within the blood itself cannot squeeze through the tight junctions, okay? That you find associated with like a normal, typical capillary. Okay, so you're getting a little bit of stuff to go through. So perhaps this is glucose, perhaps this is oxygen. Okay, so that we're fine with. Okay, so that even happens in the blood-brain barrier, right? Your brain still needs oxygen. Okay, your brain still needs glucose. But other things that we could find in our blood, we have a much harder time getting those things out because now we've also got these astrocytes that are kind of blocking most of these passageways as well, which if it's not food, if it's not oxygen, if it's not nutrients, then we probably don't want it to get to the brain. And so perhaps some of these are maybe a bacteria, okay, that would have a hard time getting into your brain and causing a brain infection, which would be a good thing, okay? This isn't 100%, right, because we still do need to get some things out of the blood. So generally lipid-soluble things are able to diffuse through this membrane and still get to the blood, right? So it's not perfect, but technically we don't need it to be perfect, okay? There are certain regions that lack a blood-brain barrier, specifically your emetic center of your medulla, which if you remember that controls vomiting, and then your hypothalamus, because one of the jobs of your hypothalamus is actually to monitor the composition of your blood to see if we've got too much or too little carbon dioxide and oxygen floating around, and so it becomes a little harder to monitor if these astrocytes and stuff are in the way. Okay, so there are pros and cons, like everything in life. Benefits: some drugs do not reach the brain. Cool. Okay. Disadvantage: some drugs don't reach the brain. So that one's kind of a double-edged sword. Sometimes we want things to get to the brain. So perhaps we have a brain tumor that we would like to get a chemotherapy drug to target this tumor. We have to work a little harder to get that drug to the tumor itself because a lot of cancer drugs cannot cross this blood-brain barrier. Alcohol, on the other hand, has absolutely no problem crossing the blood-brain barrier, and if you have ever partaken of alcoholic beverages, that does not come as a shock to you.